Hi everyone, I'm Dr. P. John Thomas, Assistant Professor in Microbiology, St. Joseph College of Arts and Science, Kadalur, Tamil Nadu. Our next session is about Roman microbiology. So what is Roman? This is a definition for rumen. Rumen is a four-walled chamber present in cattle and other animals. Animals which possess rumen is called as ruminants. So ruminants are herbivorous mammals that possess a special organ, the rumen, within which the digestion of cellulose and other polysaccharides occur through the activity of special microbial populations such as bacteria, fungi, and protozoans. So, rumen microbiology is very important for the animals. In other words, the word ruminants are animals that have a special digestive organ, the rumen, that is a unique ecosystem in which anaerobic microbes digest insoluble feed materials such as cellulose and starch and converts into energy, of course. So animals which possess rumen, the animals which possess rumen are called ruminants. So they are mostly domestic animals such as cow, sheep, and goat are ruminants. So the rumen chamber is the fermentation chamber in which all the foods are digested by degradation of protein, celluloses, amylopectin, starch, and pectin that takes place by different rumen microbes. So rumen microbes secrete different hydrolytic enzymes like proteases, amylases, pectinases, cellulases, etc., which is helpful for the digestion of the feed material. The size of the rumen in cow is 100 to 150 liters and 6 liters in sheep. So what are the different types of microorganisms present in rumen? Next we'll see. So mostly the rumen population contains bacteria, fungi, and protozoans, especially the ciliate protozoans. So without these microorganisms, the feed cannot be digested. Only the microbes play a vital role in degradation of the feed taken by the animals. So first one is cellulose decomposers. So example, Fibrinobacter succination, and fermentation end product is succinate, acetate, and formate. Next bacteria, Vibrio fibrosolvens, fermentation products acetate, formate, lactate, butyrate, hydrogen and carbon dioxide. Next one is Ruminococcus albus, fermentation products acetate, butyrate, hydrogen and carbon dioxide. Next is Clostridium species, fermentation product acetate, formate, butyrate, hydrogen, carbon dioxide. So when the animal takes the cellulose, all these cellulose decomposers will act and produce some fermentative products. Next one is starch decomposers. First factor is Butyrate ruminicola, fermentation products formate, acetate, and succinate. Next one, Ruminobacter amylophilus, fermentation product formate, acetate, and succinate. Next one is Selenomonas ruminatium, end product is acetate, propionate, succinate. And Succinomonas amylolyticus, end products acetate, propionate, and succinate. And Streptococcus bovis, lactate. So it's a very important lactic acid bacteria present in the rumen chamber. And third one, lactate decomposers, Selenomonas lactylyticus, fermentation products acetate and succinate. And next one, Megaspira elsandi, fermentative products acetate, propionate, butyrate, valerate, caporate, hydrogen gas, and carbon dioxide. Next, very important is pectin decomposers. If the animal takes a higher amount of leaves, the leaf contain pectin. These pectin molecules are degraded by the pectin decomposing bacteria such as Lactospira multipares. And end product is acetate, formate, lactate, hydrogen, and carbon dioxide. And next very important group of bacteria is methanogens. Very important organism is Methanobrevibacter ruminatium, which produces methane gas, and Methanomicrobium mobile, which also produces methane gas. Next, we'll see about what is a Rumen mechanism. So, rumen is a special digestive chamber in ruminants which provides stable, constant temperature anaerobic environment for the establishment of mutualistic associations with microbial populations. So, this rumen chamber has a higher amount of plant material ingested by an animal, provides a continuous source of nutrients for the microorganisms within the rumen, much like what occurs in a continuous fermenter. The microbial populations within the rumen can convert cellulose starch and other polysaccharides into carbon dioxide, hydrogen gas, methane, and low molecular weight organic acids. So this is the rumen anatomy. So we see here the first chamber and the largest chamber in the animal is the rumen, and the second one is the reticulum. This is the second chamber of the stomach, and third one is the omasum. This is the third and smallest chamber of the stomach, and fourth one is abomasum. This is the last chamber of the stomach. So all these digestive tract plays a vital role in degradation of these plant materials. Next, we will see about what is rumen anatomy and action. So this is the mechanism how the feed is taken by the animal and how it is digested in the fermentation in the rumen chamber, that you will see by one by one. Food enters the rumen mixed with saliva containing bicarbonate and is churned in the rotary motion during microbial fermentation. Occurs the peristaltic action grinds the cellulose into a fine suspension which assists the microbial attachment. So first the food directly enters in the rumen, and when it enters the rumen, the microbes which is present in the rumen get attached to that food material or the feed material by peristaltic action, and hereby the microbes also attaches to the food. So the food mass then passes gradually into the reticulum, where it is formed into small clumps and cuts. Small clumps and cuts is small pieces of food which is passed in the reticulum. From the reticulum, it is once again regurgitated into the mouth, where they are chewed again. So from the reticulum, the indigested food material is again regurgitated into the mouth of the animal. Now the finely divided solids is well mixed with saliva are swallowed again, but this time the material, the food material or the feed material passes to the omasum, finally ending the abomasum, an organ like more true acidic stomach because it contains a higher amount of organic acids, and here the chemical digestive processes begin containing the small intestine and large intestine. So this is the mechanism of feed taken by the animal and its digestion process. So this is the same mechanism I am going to explain here. So the food is taken, the green color shows the food or the feed taken by the animal directly enters the rumen and is fully uh, digested or fermented, and again the indigested food material reaches the reticulum. From the reticulum, it is regurgitated into the mouth of the animal. Once again it is chewed. The second chewed material again next reaches the abomasum stomach and then reaches the abomasum. From the abomasum, it will reach the intestine. From the intestine, it will convert into energy for the animal in their bloodstream. So microbial fermentation in rumen. So food remains in the rumen about 9 to 12 hours. During this period, cellulolytic bacteria and cellulolytic protozoan utilize cellulose to the disaccharide cellular bios and to form free glucose units. So the released glucose then undergoes bacterial fermentation, the production of vital fatty acids, primarily acetic acid, propionic acid, butyric acid, and the gases such as carbon dioxide and methane. So the fatty acids pass through the rumen wall into the bloodstream and are oxidized by the animal as main energy source. In addition to the digestive functions, rumen microorganisms synthesize Vitamins and amino acids that are the main source of the essential nutrients for animal. So this is an entire microbial fermentation in the rumen. So water, foodic, water, polysaccharide will be converted into small monosaccharides, then volatile fatty acids, and these volatile fatty acids will enter into the animal's bloodstream as the energy. Meanwhile, the carbon dioxide and other gases which are present in the rumen are erected by the animal and will come out of the animal's mouth. To the erectation process, the gases will come out of the rumen chamber. So 1 milliliter of rumen fluid contains 100 billion of bacteria, 10,000 fungi and 10 million protozoa. So this 1 milliliter of rumen fluid contains variety of bacteria, fungi and protozoa. So next we'll see about the dynamics of rumen. So a major feature of the rumen ecosystem is its constancy. Studies on various rumen species in different parts of the world show that most animals contain the same major rumen bacterial species, the proportions of each species varying somewhat with diet. So in addition, the natural proportions of the volatile fatty acids produced and the levels of rumen, carbon dioxide and methane relatively constant among different rumen species. The high constant temperature maintained in the rumen is 37 to 39 degrees Celsius, and the constant pH in the range of 5.8 to 7.3 by bicarbonate and phosphate. And anoxic nature of the rumen are also important factors in overall rumen function. About 100 to 200 liters of saliva per day are secreted in the rumen. So the rumen operates like a microbial chemostat. A large amount of methane gas and carbon dioxide accumulate in the rumen, the average gas composition being about 65% carbon dioxide and 35% methane. These gas leave the ruminant during erectation, or you can simply call it belching process. The next one is about the rumen dynamics. So some changes can occur in the rumen. Occasionally, changes in the microbial composition of the rumen cause illness or even death of the animal. For example, if a cow is changed abruptly from forage to a complete grain diet, so that is if you are changing the food or feed of the animal, an explosive growth of Streptococcus bovis is observed in the rumen. The normal level of Streptococcus bovis is about 10 power 7 cells per ml, quickly expands to over 10 power 10 cells per ml. This occurs because Streptococcus bovis grow rapidly on starch and grain contains high levels of starch, whereas grasses contain mainly cellulose. So being lactic acid bacterium, Streptococcus bovis produces large amount of lactate from the fermentation of starch, and this acidifies the rumen, a condition called acidosis, killing the normal rumen flora. So there is some problem can occur in the rumen if you are continuously providing one particular feed for the animal. So to avoid acidosis, animals are switched from forage rations to grain gradually over a period of few years. So if you are having animal, you have to change the feed material. You should not follow the same feed to the animal. So it may lead to some problems in the rumen chamber. So constant change of feed for animals is important to maintain the stability of rumen ecosystem. So constant change of feed is essential for the development of rumen ecosystem and as well as for the ruminants. Thank you.